Hello, my name is Nash Turley. I am a postdoctoral researcher at Pennsylvania State University in the Department of Entomology, working with Margarita Lopez-Uribe. And I am going to talk about our Pennsylvania bee monitoring program. This was a talk I gave at the Entomological Society of America meeting in what month? November 2023. And for this whole project, I'm collaborating with all these lovely people here, and you'll hear more about them. And I want to go forward. There we go. First, I want to thank the folks who have funded this research, including Penn State Extension and Pennsylvania Department of Ag, and help from many, many people on iNaturalist for submitting observations and identifying bugs. So I'll be talking about some iNaturalist data, and many folks in the Lopez-Uribe lab for help with this, and some additional BID from Rob G and Heather Hines. And then the biggest thanks goes to all the excellent people who've been helping us with the collections all over the state. Most of these are master gardeners. We have about 20 that are active now. There's been more than that have helped us throughout the last couple of years. So the two main things I want to do are to give an overview of how the Pennsylvania Wild Bee program works, and then take 2 years of the data that we've collected with our collections and compare that to observational data collected on iNaturalist from the same period of time to kind of see the pros and cons of these two different inventory and monitoring approaches. So the goals of the Pennsylvania Wild Bees is to train master gardeners and some other folks that are not master gardeners in bee monitoring methods that can be standardized and repeated over time, and to establish a continual statewide bee monitoring program, and then to use the data we collect to, among other things, update state and county checklists, study biodiversity patterns across space and time, and to track the arrival and spread of non-native species. Of course, natural history collections can be used for so many things, so there's lots of other uses to these data. So I mentioned that we are working with master gardeners. In case you don't know about the Master Gardener program, it is a program that is based out of land grant institutions, land grant universities that are present in every state in the US, and they're part of Extension. And the goal is to provide a program where regular people can go through these series of courses and ongoing education related to gardening and horticulture, and also just a lot about general biology. And they eventually get certified as a master gardener. And to continue their certification as a master gardener, it requires ongoing volunteering each year. And for a long time, master gardeners have been very excited about participating in ongoing research as well. So for our program, we've been collaborating with Valerie Sessler, who's a master gardener coordinator, and has helped communicate with the master gardener community, and helped with many other steps of the program as well. So it has not been hard to recruit master gardeners to participate in our monitoring efforts. We basically sent out emails and posts on social media for people that was interested, and we had hundreds apply, and we really struggled to find folks, to pick folks. But one of the things we did was to select participants that were spread out throughout the state, and specifically in areas that we know have few records for bees before we started. So each year we have about 20 master gardeners, and almost all of them have continued for several years now. So when folks begin in the program, we have them watch a bunch of training videos. We created these 4 lectures that are about your introduction to the ideas of bee monitoring, why this is interesting and important. We focus a lot on going through the ethics of collecting insects in general. And then through many of the intro to the methods of how to go through all the skills and techniques for collecting and processing bees. And as well as a talk just about bee biodiversity more generally. We made a number of other videos that are more focused on the exact methods that we've been using, starting with just using microscopes, using the bowl traps, putting those out and collecting them. Same with blue vein traps, on pinning bees and how to wash and dry bees before you pin them, and then using nets to collect bees. And then finally, the process of sorting bees out from a collection of insects from traps, which can be harder than it seems. It's tricky to find all those bees. So these are all present on the Lopez-Uribe Lab YouTube page, and we've had a lot of views of a lot of these, way more from all around the country and the world, especially this pinning bees tutorial, which has almost 10,000 views. So we're glad that these resources are out to the general public as well, or other researchers. So after going through all of those videos and being introduced to all those techniques, we invite everyone down to a workshop where we all come together in one place and practice all of those techniques hands-on. So we can get firsthand experience and feedback and work with each other to gain the skills that we need to do bee monitoring. And everyone then gets a kit of gear that they take home with them to do all that they need for bee monitoring, which includes collecting equipment like nets and the bowls and blue vein traps, and then for stuff for picking up the bees and then processing them and pinning them. So everything everyone needs, they get. And then these are the collection methods we've been using. They're kind of the normal things. Painted bowl traps that we put on the ground, blue vein traps, and good old netting. So kind of the heart of the program is this website and the protocols we've put together, and they're very refined and detailed protocols that go through every individual step needed. So this includes protocols on putting out the traps, on netting bees, on washing and drying bees, and pinning and labeling bees. And all of these are on the website. The URL there is on the screen, so you can check those out. We do, on all of them, we have the warning, I guess, that these protocols are not for the general public, and we're not encouraging random folks to go collect bees, only if you are part of a program where all the bees you collect are properly processed and labeled and deposited in a natural history collection. So although we are making these available for everyone, we're not just encouraging random people to go out collecting bees, which is just unethical if they're not going to end up in a collection. So after folks go out and do collections for each collection event, they go to this simple Google form that allows them to enter all of the metadata for each collection. So the date and time, the collection method, the coordinates, other things, all the things that would go in a normal natural history collection database. Each of those also has a box where they put in how many bees that they collected in that collection event, how many they've pinned. And then we print the labels using those metadata, the collection labels that go on each individual pin specimen, and mail those to them so they can add them. And they wash and dry and pin all the bees, and then they have the labels that they add to them. So eventually we get back boxes of bees from them that are all ready to go into the collection after adding a database label. So each one includes the handwritten label that they wrote in the field with all the metadata, and then the labels on the individual bees. And yeah, our volunteers have been really successful in doing a good job in washing and drying and pinning bees. We have changed a little bit in the recent years to give all of our volunteers the option to not pin if they're just like not the thing they like to do, or to not pin the small bees, because sometimes that's just a skill that some folks aren't too excited about doing. So it's always the option that we can process those bees for them. we can't go all around the state to do the collections, but we can pin and process the bees. But when they do it, it saves us, an incredible amount of time. We would not be able to process all the bees, I don't think, without all their help. So of course we have folks spread all throughout the state, and we need to get the bees back from them, which is a logistical problem and kind of difficult. But we also work with Thomas Butler, who's a extension educator, and he creates connections with other extension folks all around the state, because there's extension folks in every county, and he connects our volunteers with the extension educator, hands off the bees to them, and whenever there's a big extension event, they bring all those back to either wherever the event is or back here on campus, and then we have all of our lovely boxes of bees that we can move forward with. So once we have all those bees, we've been working, our collaborators here at Penn State have been doing most of the identification, Isabella, Sarah, and David. Of course, we've been working with some other folks to identify other bees as well. And then after that, we have all of our lovely boxes that are broken down with a unit box for each species that go into our collection that's in the lab, and then eventually into the Frost Museum, which is the entomological museum here at Penn State. Wow, they're so nice. I look at these all day. It's lovely to see all the biodiversity of bees from out Pennsylvania all organized in one place. So then there's a large databasing step, all the organization and moving bees around and identifying them and getting all the data into a database is the work of Sarah Kania, who's the lab manager in our lab. And it's just a pretty monumentous task that pretty much close to a full-time job processing and working with all of these bees. We start with our database that's in Darwin Core format. It's a bit of it takes a while to learn how all of this works, but it is the standard format for natural history databases around the world. So it's really important to start with that format, learn all of the ways in which the different columns are supposed to be formatted, which then makes it easy to upload our database through SCAN, which is a portal for insect data, which goes into global databases like GBIF. So we are committed to making all of our data publicly available as soon as possible, so they can be used for any analyses or things that folks are interested in. Okay, so that's how it all works. We've been doing this. This is our third year now, but I'm going to talk about 2 years of data based on using the collections that we did and compare that to iNaturalist data from the same period of time. So if you don't know about iNaturalist, it's a crowdsourced natural history data collection tool. There's a phone app, and you can do it on a browser-based place as well. Go out and take photos. You need a time and coordinates for those photos, and then you can upload it. It has pretty amazing robot tools, AI tools that can get a pretty good guess of the identification of stuff. And then they're sometimes wrong, but normally they do pretty good. And that can give an initial identification, and then other folks in the community can go through and agree or disagree on that identification. Once there's 2 people that agree on identification, it's a little more complicated than that, but we have sort of multiple people agreeing on that identification. It's called research grade, as long as it also had coordinates. And then that's a way of kind of filtering out ones that never got identified or people disagree on the identification. So for the data I'm showing, it's only filtered by the ones that are classified as research grade. Still doesn't mean they're all right. You could just have 2 people agree and it could be totally wrong, but we're just kind of taking the data for what they are. So one of the questions we can ask is, can a photo-based observation approach like iNaturalist replace the types of collections we've been doing, or do they complement each other? So in that time, our collections got a little over 9,000 bees. And in this map, you can see the black dots are individual collection locations, and the shading of the counties is representing the number of specimens that were collected in that county. So we're spread out throughout quite a bit of the state, but it's still kind of patchy based on where we have folks that are doing collections. So in that same period of time, there were about 7,400 observations on iNaturalist. You can see there's a lot more individual collection locations. And it's spread out as well, but very clearly clustered in the cities. It's not too surprising. There's more people there, so that's where there's more data collected. So the first comparison is looking at just the total number of species that were collected or that were observed or collected in those 2 years. So with our collections, we got 243 species, which is over half of the species that are known to be found in Pennsylvania. That's about, so far the checklist has about 430 species. And that's in 43 genera, which is nearly all of the genera that are known to be in Pennsylvania. In iNaturalist, there's 85 species from 33 genera. So a breakdown of what species were in each approach. We had 169 species that were unique to our collection. So the big blue part of that circle is species that only we found that were not found in the iNaturalist data. In the middle, there are 74 species that were in both collections, and then there were 11 that were unique to iNaturalist that we did not find in our collections. So the next thing I did to be able to really do some quantitative analyses was to take 13 counties of data where there was over 50 bees observed or collected in each data set. So we caught over 50 bees and iNaturalist had over 50. So I looked at biodiversity things as well, and it was similar results to the overall. There was over twice as many species at the county level in our database. And the next thing I wanted to do is look at the body length of species to get a sense of the biases in these different data collection methods. So I created a database of the body length of every species in Pennsylvania, and then I used that to look at the average body length of the species that were documented in each data set. So each point here is a county and the average length of species that we found. So the body length of bees was 1.4 times larger in the iNaturalist data set. It's not a particularly surprising result. Of course, if you're going out and taking photos, it's easier to spot big bees. It's probably easier to get photos of them. And also bigger bees are more likely to get identified to the species level and end up as research grade observations. So perhaps not surprisingly, there's a pretty strong bias towards larger bodied species in iNaturalist. We also did get the large bodied species, but because of so many small ones, the average is much lower in our collections data set. So similarly, did the same type of analysis, but instead this case, whether a species was categorized as non-native. We have over 30 non-native species of bees in Pennsylvania, and tracking those is one of the goals of our program. So we found that there was over twice as many relative number of species that were non-native in the iNaturalist data set. Of course, this is a percentage. So because there was a lot more species in our collections, we documented about a similar number of species, but relatively there's a bias towards non-native species in iNaturalist. Who knows why? The folks are collecting more in cities. These species are associated with humans. A variety of reasons. So one of the ones is photoed here is the European wool carder bee, which is this aggressive bee that you often see in gardens, and it chases around other bees. So, but it does suggest that there's this bias towards non-native species, which could be seen as an asset to iNaturalist if one of your goals is to track non-native species. So in conclusion, we've really found that our hybrid approach is really great. So what I mean by hybrid approach is that we do collections, and we collect high quality research grade data with our collections, but we're also collaborating with folks from the public. So it's this mix of high quality data, but working with the general public to collect the data, or a small subset of the public. And working with master gardeners in this process has been really excellent. You know, it's necessary to have folks that are enthusiastic about participating in research and willing to put in kind of an incredible amount of time and effort, just because it's been, from what we've heard from them, very rewarding and interesting to be able to really collect data that's super high quality and we can learn a lot from. So we're just a fan and advocate of working with master gardeners because they're just a very unique and dedicated crowd. So I recommend it. And by comparing our data to iNaturalist, we find that doing lethal collections is collecting a vastly more biodiversity than photos. Again, that may not be super surprising, but we do need to be at a point where we need to justify collecting and killing a bunch of bees for research. And we are finding a lot of unique discoveries and a lot more biodiversity captured. So if our goal is to do biodiversity monitoring and updating checklists and things, we do see that the photo-based observation method is just going to be inadequate for those particular goals. However, iNaturalist does clearly show a very good strength for documenting large-bodied bees, which could fit very well into a lot of goals for programs like monitoring bumblebees and things, or comparing how these bees are performing in different habitats. There's all sorts of research programs that we think could be very well, you could accomplish a lot of research goals using iNaturalist, but it's just not particularly good for like overall biodiversity monitoring. And we also see this bias towards non-native bees, which could be seen as a plus if that's something you particularly want to study. And finally, as just sort of an overall conclusion, one of our bigger goals for monitoring bees is that eventually this will play a role in conservation or even policy. So one of the things we're working on is using our data and other data to do NatureServe rankings of different species within Pennsylvania. We also want to look at patterns of biodiversity, or maybe biodiversity hotspots, which could give insight into managing of different places or prioritizing conservation efforts. We're still kind of new to all of this, but in the longer term, we do hope that all of these extra biodiversity data, this basic natural history data, will have conservation implications as well. So that's all I got. Thank you for listening. If you want to get in touch with us, have any questions about how we're doing all of this, get in touch with me or Margarita or Sarah. All of our materials and protocols, everything are on the website here. We're glad to, we hope that maybe something we're doing, if it works for other states, could be a program that could be replicated, and we're glad to help or provide any of our resources to make that possible. That's it. Bye.